Good morning, and welcome to Science with Kate. Today I'm going to teach about what made you you, and that all began with the process called meiosis. What is meiosis, you may ask? Well, meiosis is a modified type of cell division in sexually reproducing organisms, consisting of 2 rounds. That's right, 2 rounds of cell division, but only 1 round of DNA replication. It results in cells with half the number of chromosome sets as the original cells. Let's take a closer look at this. Wow, that looks really scientific. Let's start somewhere else. Oh yeah, that's what I'm talking about. What this donut is doing such a wonderful job depicting is the very first step in meiosis, called prophase 1. Prophase 1 is where it all begins, ladies and gentlemen. You got your chromosomes condensing. You got your homologous pairs matching up and crossing over. What is crossing over, you may ask? Well, I've got the answer for you. Crossing over is the exchange of corresponding segments of DNA, molecules by non-sister chromatids. Oh hey, would you look at that? Here we have a nice picture of prophase 1 that demonstrates points of crossing over at the chiasma. Not only that, but you can also see centrosome movement in the beginning of spindle formation. That's enough of that, don't you say? Let's move right along into metaphase 1. Holy deliciousness! Here at metaphase 1, we can see our homologous chromosomes arranging themselves on the metaphase plate. You know, where all those delicious red, green, and blue sprinkles are lined up? Each sprinkle, or I guess I should say homolog, is attached to a kinetochore microtubule. Each microtubule will run to one pole or the other. And with that, it's time to say goodbye to metaphase 1 and hello to anaphase 1. Show me the donut, Kate. Would you look at that? Here at anaphase 1, we'll see the sprinkles, well, the homologs, move towards the opposite poles while being guided by their spindle apparatus. It's important to note that this is the phase where the homologs separate and become sister chromatids. Alright kids, we're almost done. We've just got one phase left in metaphase 1, and that is telophase 1 and cytokinesis. Let's go take a look. Oh my holy mother of donuts, doesn't that look good? In telophase 1, each half of the cell has a complete haploid set of replicated chromosomes, being composed of 2 sister chromatids. Here we can also see the cleavage furrow associated with cytokinesis. Once these processes are completed, 2 haploid cells will be formed. Or in this case, 2 donuts, which who doesn't need more donuts?
And that's meiosis 1 for ya.
Wasn't that riveting? Now it's time for meiosis 2.
You forgot there are 2 steps, didn't you?
Luckily for you, meiosis 2 is a lot like mitosis. I know those donuts probably made you hungry, so this time I used gummy worms instead. In prophase 2, a spindle apparatus forms, and chromosomes, still composed of 2 chromatids, associated at the centromere, start moving toward the metaphase 2 plate. And that's about it. Now it's time to move on to metaphase 2. Would you look at those sister chromatid gummy worms? Here, in metaphase 2, they will position themselves on the metaphase plate as in mitosis. Additionally, the kinetochores of the sister chromatids are attached to microtubules extending from opposite poles. It is important to keep in mind that due to the crossing over that occurred in meiosis 1, the sister chromatids are not genetically identical. And there you have it. That's metaphase 2. Now it's time to move right on along to anaphase 2.
Let's look at some more gummy worms.
Oh, nom 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 nom. In anaphase 2, the sister chromatids separate due to a breakdown of the proteins holding the sister chromatids together at the centromere. The chromatids will move towards opposite poles, and soon the cell will enter telokinesis 2 and cytokinesis. In telophase 2, the haploid daughter cells will form. In a result, you will end up with 4 haploid daughter cells that are genetically distinct from the other daughter cells. This is complete. Pretty easy, hey?
I could have done that in my sleep.
Thanks for watching this episode of Science with Kate, and I hope to see you next time.